On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Precision for Medicine, I'd like to welcome you to Meeting the Clinical Utility Needs of Payers and Regulators for Molecular Diagnostics. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. At this time, I'd like to introduce our presenters for today's webinar. David Parker, PhD, is Vice President of Market Access Strategy for Precision for Medicine. David has been active for more than 25 years in the healthcare industry, including 16 years of strategy consulting experience centered on the intersection of reimbursement, health economics, clinical and regulatory science, and marketing strategy. He currently leads Precision for Medicine's Market Access Consulting practice and provides strategic advisory services to the firm's biopharma, device, and diagnostics clients. His consulting encompasses all aspects of reimbursement, market access, and evidence development strategy, with a particular focus on personalized medicine, molecular and advanced diagnostics, and medical devices. Throughout his career, his work has resulted in numerous successful product launches, guided major investment and acquisition decisions, and driven favorable coverage and payment determinations by public and private payers alike. David received his PhD in Cell and Developmental Biology from the Massachusetts Institute of Technology, where he was a National Science Foundation Fellow, and received an AB from Princeton University. Our second speaker is Judy Smith, Master of Science and Medical Technologist, American Society of Clinical Pathologists. Judy is the Vice President for In Vitro Diagnostics, Regulatory, and Quality for Precision for Medicine. She works with Precision for Medicine's Translational and Regulatory Sciences practice, which was established to provide value-added scientific and regulatory solutions for development, marketing authorization, regulatory compliance, and stewardship of innovative healthcare products. Judy has worked in the medical products regulatory, quality, and clinical trial areas for over 30 years. In that time, she's been responsible for the regulatory submissions and approvals of a multitude of companion diagnostic assays, in vitro diagnostic products, and biological in vitro diagnostic assays. She has designed and implemented clinical trials for IVDs that were used in regulatory submissions. She also is on the faculty of the Raps University as an IVD subject matter expert. As a member of the FDA Industry Roundtable, Ms. Smith was awarded the FDA Group Recognition Award. She has served as president of the Association of Medical Diagnostics Manufacturers since 2006. This organization is dedicated to the in vitro diagnostic industry and provides educational programs with the FDA CDRH Office of In Vitro Diagnostics and Radiological Health. Ms. Smith received her Bachelor of Science from Temple University and her Master of Science from Medical College of Pennsylvania. With that, David, I will turn the presenter ball over to you. Welcome.
Thanks very much, Elizabeth, and welcome to our webinar this afternoon. Judy and I are delighted to be able to present this discussion, and hopefully we'll have an opportunity to take a number of good questions at the end and get into some specific details for you. So without further ado, today we're going to cover a few areas. The first thing that we want to do is really present our hypothesis. We're really trying to prove here today. Then we'll talk a little bit about clinical utility definitions that different stakeholders hold. We'll talk about evidence requirements of the FDA and third-party payers, commonalities of clinical utility evidence development, and then we'll go into some specific study design efficiencies that might be available to people who are doing dual-purpose studies, as we like to call them. Our hypothesis is really the following. First, clinical utility evidence development is really the most expensive part of diagnostic creation and development. Oftentimes, the laboratory program for developing the diagnostic is, in fact, much more constrained in scope than actually the clinical evidence development necessary to gain either or both of FDA approval or to gain reimbursement from payers. And in fact, those two entities are the two main access gatekeepers and the two main consumers of clinical evidence. That is to say, the FDA and third-party payers. Their evidence needs may differ and in many cases appear to differ, but what we have found is that with appropriate foresight and appropriate planning, you often can address those evidence needs simultaneously in a dual-purpose study. And in fact, finding and taking advantage of those synergies is the key to cost-effective clinical evidence development in diagnostic test development, where in fact return on that investment is critical because the markets, of course, are so much smaller than they are for pharmaceuticals. And in fact, Jeff Shuren, who you all probably know and many have interacted with, was quoted just last year, and I'll actually go ahead and read this to you, where he says, there have been cases where the payers heard what we're planning to do for a study, and they say that maybe with a small tweak, that clinical study would meet their needs. In those circumstances, it's not requiring much effort on the part of the sponsor, but they save themselves from having to do a whole new clinical study for getting the payer support. Okay? That's our premise, really, and we have actually found that to be true. So, the first step in really understanding the possibilities for dual-purpose studies are understanding how key stakeholders define clinical utility. And I won't read all of these words, but suffice it to say that it's really important to actually look at the critical stakeholders, whether they are payers, specifically, represented by the MolDX program, whether it's the agency represented by CDRH, or third parties that might be developing guidelines, and to really actually dig in and see how do they define clinical utility? What are they actually going to be looking for to be demonstrated in these studies? And in fact, understanding how those different stakeholders' definitions overlap enables the design and the execution of these dual-purpose clinical trials. Evidence needs, then, are sensitive to the value perspective. And in that context, the notion of safe and effective, as the FDA looks at it, blends into useful as payers look at it. And so looking at how payers view evidence for diagnostics, they're looking, for example, for objectively improved clinical outcomes, difficult to establish for diagnostic tests because, of course, the improved outcomes depend on the therapeutic efficacy as well. They're looking for things like changed physician treatment behavior, as long as that changed behavior is to an accepted or a better alternative, that is well understood to be a better alternative. Or they may be looking for changes in referral patterns, where, for example, you could demonstrate reduced numbers of referrals from a primary care provider to a specialist, or referrals to a more appropriate specialist. Or they're looking for the test to guide the same or better clinical results, possibly at a reduced cost because of avoided other diagnostic tests or because the therapy is more effectively identified.
Okay, and from the FDA perspective, their evidence is based on understanding the safety and effectiveness of the product. For IVD products, the safety, we tend to think of safety as, are we going to get electrocuted if we touch this instrument or this device? For IVDs, it goes beyond just the equipment safety. There's also, the FDA considers the assay performance itself to be part of safety. For example, what's the impact of a false negative or false positive on patient treatment, patient treatment or diagnosis? And the same goes, say, for a quantitative test where you've got a high positive or a high value or versus a low value, and is that low value incorrect? So those are the kinds of things the FDA is looking for when they're looking for safety proof. And the effectiveness is very simple. It's, does the assay do what the label says it does? Does it quantitate an analyte? If you're claiming it's going to be suitable, it drives therapy selection, is it going to be able to do that? And so that's how you define effectiveness And you can see it's a broad range of possibilities based on the claim you want to make. The only things that I'll say, though, is unless you're going to claim a clinical outcome such as better patient outcome, better health, faster treatment, the clinical outcomes are usually not within the review arena of the FDA. And likewise, cost evaluation is definitely outside their purview. Once we understand these balances and try to blend them, all this reduces down to two questions, one from the FDA and one from the payers. And from the FDA is this test fit for purpose? And from the payer, is the purpose useful enough that we should spend our money on it? And those are the two key questions. Now, how do we answer the fit for purpose question to be able to figure out what is required to generate data for the evidence? And of course, as in any regulatory question, the answer is it depends. And it depends upon the level of risk of the assay. So there are three mechanisms, regulatory pathways, and each of those regulatory pathways have different requirements. The FDA de novo and the PMA, the F10K de novo 510K and the PMA. So in the 510K, as we all know, it's a demonstration of equivalence to a previously cleared product. Analytical validation is important. Clinical validation is really in the form of clinical sample studies. In other words, you're using human samples from that population, but you're not actually going to the patient and enrolling them in prospective studies. The only time you might need that kind of evidence is if you are proposing a new population or a new indication for an already cleared product. De novo is a little bit, it's kind of somewhere between a 510K and a PMA. It's the same process and the same information as in a 510K, but the clinical patient study to develop the evidence is probably likely to be required because it's a new product or a new claim. And at the high end of the risk factors is the PMA, and that requires evidence of safety and effectiveness for the population and the specific use. There's a definition by the FDA that a device is effective when it can be determined based on valid scientific evidence that it will provide clinically significant results. The valid scientific evidence used to determine the effectiveness of a device shall consist principally of well-controlled investigation. So that's where they're asking for the types of studies that we're focusing a little bit more in this talk. So the question is, we know now that there are differences and similarities between what the FDA requires and the third-party payer requires, but it's not always possible or practical to run a dual-purpose study. So we begin to ask the question, when is it suitable? And to answer that question, you have to kind of look at both pieces of it, the FDA piece, the payer piece, and evaluate the type of products they are. So that's what this slide represents, our thought process, how you walk through the concepts of identifying where the overlaps lead to a more efficient trial. So the first category is the FDA risk level. And we all know they're low risk, moderate risk, and high risk. The low risk just requires QSRs and doesn't require a submission, usually. A moderate risk has a 510K or a de novo 510K. And the high risk has the PMA. So we now know what the FDA risk levels are. We then overlay that with the payer cost impact questions.
So payers, as many of you know, although of course they have multiple interests in the utility of a test, payers oftentimes reduce that really to what is the cost impact to them, what's the budget impact of that test, and or the therapy that's going to be guided by that test. And at the risk of oversimplifying a little bit, in our algorithm here, we've reduced it to low cost impact and high cost impact. Of course, there's many shades of gray in between that might drive the need for payer evidence and then, therefore, the feasibility and desirability of a dual-purpose study. But in this simplified model, we'll just separate it into low and high. Well, if you look on the far left-hand side of the screen, what you'll see is that for low-risk tests, you could either have low or high-cost impact. Now, in practice, a low-risk test, which does not require even a 510K, is unlikely in practice to be either expensive or directly guiding any sort of expensive therapy. And so in practice, it's really probably unlikely that a low-risk test is ever going to have a high-cost impact for a payer. With moderate risk, of course, it could go either way. There are moderate risk tests which might be very meaningful from a clinical treatment algorithm perspective or a diagnostic and treatment pathway perspective. That could go either way. High-risk tests are probably high risk because they are really guiding, if you will, life and death or decisions or clinical decisions that have a high therapeutic consequence. High therapeutic consequences or life and death clinical decisions very often have a high cost impact for the payer. And so in point of fact, you're likely to see more of those high risk tests actually having a high payer cost impact. But then if you combine those and actually look at what's the need for clinical evidence, again, starting from the left, low FDA risk, low cost impact, you're unlikely to need any clinical evidence whatsoever actually to bring that test to market. And in fact, appropriate coverage and smooth reimbursement is not likely to be a problem for such a test. If you have high cost impact but low risk tests, the FDA probably is not going to require clinical evidence, as Judy's already said, but payers very likely will. And so you're looking then at a clinical utility study, but not actually a clinical approval study for regulatory purposes. In that middle category, moderate risk tests, low cost impact, as Judy mentioned, FDA might or might not require clinical evidence. Payers almost certainly would not. The three categories to the right, moderate risk, high cost impact, or a high risk test, really with any sort of a cost impact, almost by definition of what high risk entails, you're going to require evidence for both FDA and payers. And so if you look at these three areas, the three categories to the right really offer possibilities for clinical study synergies that you could achieve.
Once you've decided that your product assay is good, kind of makes sense to do a dual purpose trial, you begin to design that trial. What will that trial look like? And you first ask the regulatory questions, and which is, what is the intended use? Is it going to be a screening test? Meaning you are taking apparently healthy individuals and running the test on them? Or are you going to have it as a diagnosis, which is meaning that you've got a patient with signs and symptoms of some condition and you're using your test to diagnose? Those are two different levels of questions.
And I might add they're very different for the payer perspective as well, especially because a screening population is obviously going to be much larger than a test indicated for diagnosis, where you've already narrowed the population by virtue of the signs and symptoms you just mentioned.
Yeah. And then there's prognosis, which is predicting future events. And the fourth category is treatment selection. So what's the intended population? Is it going to be asymptomatic individuals? Is it going to be patients with signs and symptoms? Is it going to be mid-treatment, such as they're in the process of being treated and this marker is going to change their treatment? Is it specific subgroups within the above? within the above categories I mentioned, such as it's only those patients with a condition who are positive for a specific marker. And then what are the endpoints? Is it correlation to a diagnostic gold standard, which by definition there's no such thing as a kind of a one gold standard for diagnosis. It would be a series of tests or procedures to define the clinical truth of the patient. Determination of disease risk. Is that what your claim is going to be? Is that your endpoint, that you are going to look for risk of recurrence or risk of some event as one of your endpoints? What's the likelihood of recurrence? Once again, that's another one where you would be looking, that recurrence would be the endpoint that you're looking for. And treatment segmentation of the population, is this marker going to tell us that this drug is better than that drug? So those are the kinds of questions. And answering these questions, both from the FDA perspective and then looking at it from the payer perspective, which may give you the same or slightly different answers that will help with the clinical trial design to make sure that you meet both needs.
So then if we look at beyond the FDA's requirements for safety and efficacy, we can begin to ask a number of questions that help us align potential study outcomes with payer perceptions of clinical utility. And you'll immediately be able to see how many of these questions relate back to the categories of evidence and the endpoints and the intended uses that Judy was just mentioning. For example, if you look at the type of study outcomes, mortality, morbidity, is it intended to quantify how much morbidity there is? There are diagnostic tests, for example, that are designed to provide peace of mind to a patient. You know, these might be prognostic tests that don't directly guide a patient into alternative treatment pathways, but instead simply reassure the patient that they are likely to be disease-free in the future. Well, payers will look at those in different ways because from their perspective, they have different clinical values. Timing. You know, when would the clinical benefit be realized from the diagnostic information? Now, this might seem to, not have a lot of impact on how a payer would actually look at things, but in fact, if the clinical benefit is based on, let's say, a 5-year course of therapy or maintenance therapy that's guided by the diagnostic, well, that payer might well be thinking in terms of what they call disenrollment, where there is going to lose the benefit, the downstream benefit, because the patient has changed jobs and they've moved on to another plan. Particularly relevant, of course, in this era of reduced restrictions on prior conditions. So it makes a difference. Could it be so long before the benefit is seen that it's unclear what really caused it? So let's consider, for example, a molecular diagnostic test that could indicate you should change lifestyle, okay? Well, if I do, in fact, cut back on butter in my food because I've got a cardiac test which indicates I have a predisposition to myocardial infarction, and it turns out that if I'm really adherent to cutting back on butter, I have a lower mortality of the population 10 years down the road, could anybody really say that that diagnostic test led to that benefit? That's a question that payers are going to ask. How tightly linked are the test and the benefits that appear to be gained from the test? And so that actually segues directly into likelihood. How directly does the test relate to the projected benefit? Or are there multiple independent intermediate stops on the patient journey between the test and the benefit gained? Can something beneficial be done for the patients that the test identifies? Well, for a very, very long time, I give you the example of HPV molecular testing, where for quite a while after the introduction of the first HPV molecular test, nobody knew what to do differently for the patients. Should we watch and wait? Should we increase the interval? Decrease the interval between pap smears? Should we do colposcopies? How frequently should we do it? Nobody knew what to do. And so the utility of the HPV molecular test at the start was unclear. Of course, it's very well accepted now. It's known what it's good for, but it actually took quite a few years of clinical experience to understand what that utility might be. In terms of the cost, of course, we all know payers are acutely interested in cost. So how much does the test itself cost? The level of payer scrutiny is going to be different. The level of evidence required is going to be different for a test that costs $100 versus a molecular test that, as many do these days, might cost $1,000 or more. Are the downstream or dependent costs increased or decreased? So a test that itself might be relatively low cost, but whose desired outcome is increasing, let's say, a more interventional diagnostic test, an angiogram, for example, or a CAT scan, that might actually be viewed as additive to the cost of care overall. And for some payers, and particularly as you turn your attention outside the U.S., you get more explicitly the question, is this test cost effective? And payers care about that, and they want to see endpoints and measures in the clinical studies that actually give you an assessment of cost effectiveness. With those kinds of considerations in mind, you can say, okay, let's look at the FDA and third-party payers and what kind of relationships do we actually see between FDA study design and third-party payer measures of clinical utility. And we've got here on each side, obviously not all of the different kinds of endpoints or factors that you might look at, but ones which are particularly meaningful for CDRH on the left, and others which we know from experience are particularly meaningful for payers. And so, CDRH, the FDA, target sample type, analytical data, stability, interfering substances, assay range, target population, claimed use, diagnostic, prognostic, clinical impact. Is this a companion diagnostic? Is this a PMA IVD with no companion diagnostic claims? Is there labeling for treatment and the like? On the payer side, there is interest in analytical validity and clinical validity. There may be interest in the reduction in the number of diagnostic tests. They may be interested in understanding whether or not this new molecular test is a replacement for higher risk or more expensive procedures. Is there a reduction in the time to diagnosis? Is there better prediction of therapeutic effectiveness and side effects? So at first glance, these two rosters of factors that the FDA and payers might care about look to be very different. But if you dig a little bit deeper, you begin to see parallels. So target sample type is a component of analytical validity. Similarly, the analytical data is a component of analytical validity that payers care about. Clinical validity ties directly to the target population. Now, the payer will want to know how frequently will this test be done and in what sorts of patients for what sorts of indications. They will similarly be interested in the claimed use as a component of clinical validity because that will be the lens through which they look at the results of the test in terms of whether or not they deem it clinically useful. Okay? And they'll have very different standards, let's say, for a prognostic test than they would for a diagnostic test. So clinical impact, that could tie directly, for example, to payers' interest in the reduction of the number of diagnostic tests performed. Similarly, the payer's interest in a replacement for a higher risk procedure would tie directly to clinical impact. Also, reduction in time to diagnosis could be a clinical impact that the FDA would want to see demonstrated in the studies, as could better prediction of therapeutic effectiveness and side effects. And finally, labeling for treatment or labeling as a diagnostic test or a companion diagnostic test would be tied directly to payers' interest in better prediction of therapeutic effectiveness. So if you dig, sort of scrape back the surface and dig one layer deeper, you begin to look at how these two kind of rosters of endpoints, if you will, tie together and where you should be making these linkages in the study design itself. Okay, so I don't want to belabor this because Judy's got a number of more specific examples that we'll dive into shortly. But again, if you look at the FDA and you look at payers, the evidence needs and the potential utility endpoints that we have in that top row would be very different potentially for a hypothetical. In this case, we've postulated a hypothetical risk prediction test that's based on specific pathway genomic markers or gene expression. And so you look down those two lists, and the FDA is interested in quite different things from the payers, apparently, with maybe the exception of health outcomes. And so if you were to consider each of those stakeholders' evidence needs independently, then you could come up with any number of different study designs that, let's say, independently could meet the FDA's evidence needs, as, for example, a retrospective study of the relationship between a CDx marker and the risk of pathway-specific adverse events, or, a medium-term prospective interventional study. Similarly, if you looked at the payer's concerns, independent of the agency's concerns, you might come up with a number of different clinical utility study designs. A long-term high-enrollment prospective observational study, as an example, or, as another example, a medium-term prospective interventional study. It's when you begin to look at them simultaneously that you then sort of say, you compare and contrast those alternative study designs, and you look for ones that might actually be able to meet the needs of both stakeholder groups. And in fact, in this particular hypothetical, that could be the medium-term prospective interventional study.
Now, these next three slides are actual examples of types of studies you could consider for addressing the clinical evidence needs of the FDA and the payer. These are purely examples, and what we did was we started with the least likely to be good for a dual purpose, and we moved to the more likely. And what we've done is taken the concepts that we have just spoken about for the last half hour or so, and now put them into practical on paper what a trial might look like. And each of them, by the way, from an IVD perspective, each of these examples gets a little closer to a dual purpose study based on where the marker shows up within the study design. And that's why I've color-coded the marker test in red, because that's kind of where you begin to, when you see where it keeps showing up relative to the rest of the testing, you can see where it changes the type of data you can generate and the types of clinical evidence, therefore, that you can get for the payers and for the FDA as well. So this first example is you enroll the patients and you randomize them to a treatment or a control group. And with each group, you go ahead and run the marker on each of them. And then you look at the test positive and the test negative within each group and look at their performance compared to an investigation, the standard of care versus the investigational group. Now this can be a retrospective study because you can, and a lot of companies run it that way, where they go to a biorepository and purchase the samples with the clinical diagnosis and the outcomes of the patients that have been followed for 5 years, for example. So you have those specimens. You already know what the treatment looked like if you're going for that treatment piece, what the patient's outcome was, what treatments, other treatments they had. You know the history of those samples. And then you look at the marker and say, would my marker have seen a difference in the test positive and test negative groups in the investigational group versus the control group? So the study can be retrospective, and it provides a correlative relationship of the test to the treatment. So, for example, you can look at it and say, was my marker predictive, since I followed these patients, or I have the data on these patients, was my marker able to predict the recurrence of this disease? But in this design, there's no actual change in treatment. There's no effect on the treatment. The marker did not change the treatment possibilities or impact the clinical outcome of the patient. So this is more of an early proof of concept study, or one where you are not really generating third-party payer clinical evidence. So this is more an FDA type study. The second example that we'll go to, you'll see that the marker is the first step in this randomization process in this test, so that you take the marker, you enroll the subjects who meet your inclusion/exclusion criteria, and you randomize them by test results, which really isn't randomized, come to think of it. You put all the test negatives in the standard of care group or control group, and you put all the test positives in the new treatment group. And then you look at patient outcome. And what you're looking for is efficacy in the standard of care group and separately in the new treatment group. Now, this study is prospective, and it evaluates, you can get evaluation of the response rate of the new treatment test positive subjects versus the standard of care test negative group. So what that will give you is some indication that in the test positive group, the new treatment, for example, had a response rate of some value compared to the standard of care. So that gives you some, not only FDA data, but it also gives you some third-party payer data. The only problem with this is there's no evaluation of the new treatment on the test negative patients. So you really don't know in this group whether it was all the benefits were all due to the drug or due to the fact you were able to identify them as positive. So this study will work for both third-party payers and FDA if it's a well-characterized marker. If there's been lots of literature, such as HER2, if you know that HER2 or a similar type of marker where there's been lots of evidence about it, and you're not in that early detection phase, and your treatment is targeted specifically for that SNP or what have you, that the test diagnosis. Now, the third one is another example that does address both FDA and third-party payers. This is one, and by the way, this is just purely an example. You actually have, it's a dual testing. So you've got the perhaps already cleared or approved diagnostic procedure, and you run the marker along with it. And then you look at the algorithm of results from both tests. So your options are that your screen, in a screen normal population, you've got either a positive or negative marker, and in the screen abnormal patients, you've got positive and negative markers. So you can start to look at standard of care versus a new treatment. You can start looking at within the screen normals. The screen normal, for example, the screen normal, the marker positive, you would do increased follow-up. Screen normal, marker negative, you would do just standard follow-up, and you would watch the patient and look for outcome. And that gives you some interesting data about, in a normal screen, what the impact is of the marker. And the same with the abnormals, in the marker negative and marker positive. If in the marker positive you have a new treatment in there, then you can begin to look even more closely at the differences between standard of care and new treatment, and you end up with even more data. We got a question about what do I mean by abnormal. I don't mean abnormal patients. I mean the screen test shows that it's not I was trying not a negative. For example, in a mammogram for breast cancer, you're either negative or it's diagnosed as an abnormality, so that you're not making a judgment call on it, so then you do further follow-up. So that's what I meant by abnormal. Anyway, so in getting back to this just very quickly, my point was the test is prospective. It compares patient outcomes across groups. It generates a lot of useful data to address both the FDA and the third-party payer. It answers fit for purpose, and it also answers purposeful use.
Just to sum up then, what we hope we've been able to do is to really sort of provide a study design mode of thinking. I don't know that I'll call it anything as rigorous as an algorithm, but at least a way of thinking about evidence needs of your two most critical stakeholders in a way that, if done right, can allow you to meet both of their needs with the same study, actually, not even two successive studies. One thing we want to make clear is we're not providing a recipe for, the way to do a dual purpose study. There are multiple ways to do it, and in fact, that I think is a critical takeaway, is that you really need to understand the specifics of the test that you're actually designing the clinical study for. You need to understand the specifics of the disease, the test's intended use, the therapeutic pathway, and the FDA and payer implications of all of those for both risk on the payer side and potential cost impact on risk on the FDA side, and potential cost impact on the payer side, and look at those holistically in order to design the most cost-effective study that you can design. And we've seen this. This is possible. We've done this, and we've seen other companies do it as well. So this is, I think, clearly a way in which you can increase the cost effectiveness of your clinical development program with the appropriate planning ahead of time. And with that, I would like to turn the presentation back over to Elizabeth, who I think has queued up a number of audience questions that have come in while we've been speaking.
Yes, I have. We have a pretty good number of them. A couple of them are related to CLIA, so let's start with those. The first one is, will laboratory-derived tests that are developed in a CLIA lab environment be covered?
Well, since it refers to coverage, I'll actually take the lead on that. Judy may have some other comments as regards FDA regulation of LDTs or laboratory-developed tests. So the short answer is that payers are pretty agnostic whether, at present, they are pretty agnostic whether or not a test is a laboratory-developed test or an FDA-approved test. I think it's safe to say that by now payers have seen examples of FDA-approved tests that have no perceived payer utility, clinical utility, and conversely, laboratory-developed tests that have robust clinical utility. So there's no obligatory linkage in the payer's mind for coverage on the basis of clinical utility, what kind of test it is. Now, having said that, I will say that there is some evidence, particularly in the MolDX program, that many of you are familiar with, which is the program used by Palmetto and likely, I think, in the future by all of Medicare to determine which molecular diagnostic tests will be covered and reimbursed. There is some preference that's being established for FDA-approved tests. And in fact, in some conversations that I've had directly with Elaine Jeter, she has indicated that that is at least one of the reasons why she has been eager to have these tests separately identified with the individual Z codes or Palmetto test identifiers, is that she does in fact see a difference between an FDA-approved version of a test and a laboratory-developed version of a test. And she might, based on the evidence provided, wish to have differential coverage or maybe even differential payment for these tests, and therefore needs to have them identified separately.
Okay. I think the next question might be more relevant to the CLIA, and we'll address both comments together. Elizabeth, do you want to read that next one? Or do you want me to?
I can read that. Next-generation sequencing-based tests for targets like BRCA can be developed in a CLIA lab, immediately sold, and are reimbursed without any interaction with the FDA. Do you see the FDA stepping into the regulatory process for next-generation sequencing-based tests and perhaps requiring clinical trials?
Well, the first part of the question is the million-dollar question. We're all waiting to see what the FDA is going to do with regulation of LDTs in a CLIA lab. I would say probably some type of regulation will occur, but what I've heard is it'll be risk-based. So if you've got a marker that is diagnosing cancer and recommending specific treatment, you're probably going to have to visit the FDA. If it's prognostic, the FDA may consider that to be low enough risk that they wouldn't require a submission of any type other than registration and listing. So I think in general terms, that's what I'm hearing. Next-gen sequencing-based tests, I guess it depends upon what the claim is going to be. If it's a claim for aborting a fetus, well, you're definitely going to have to visit the FDA, in my opinion, when they get to regulating it. If it's determining treatment for cancer, then I think that the FDA will be looking at that too. Now, as requiring clinical trials, there is a CLIA requirement that you validate the assay, and that you perform clinical validation as well as analytical validation. So there is some clinical testing or some type of clinical trial, at least, that you're going to have to run just for the CLIA piece. In my opinion, you're also going to have to run it to be able to market it to the doctors to order, because if you can't demonstrate that this is useful in diagnosing the patient or early diagnosing the patient or designating the appropriate treatment, you're not going to necessarily have a large following of doctors ordering it. So I tend to think the clinical trials that you're going to need, whether you're CLIA or FDA, if it's a new marker, from a business perspective purely, is to get publications showing it's worked in these different populations so that you've got the evidence to prove it and communicate it to the world. So I hope that's answered your question.
Oh, we have kind of a follow-up question that came in while you were answering that one. What if the test was developed in an ISO 13485 certified laboratory?
I would say if it's in the United States, if it's a U.S. lab, it doesn't matter whether it's CLIA or ISO, it's still going to require some kind of FDA submission if it's diagnosing critical diseases or guiding critical treatment. If the lab is outside the United States, then it becomes a question of if our U.S. samples going there and information being sent back to the U.S. doctor for treatment, and I would say, yes, the FDA would probably visit you then too. Whether you have to run validation for it, I think both CLIA and the ISO guidances, standards, are pretty clear that you have to have some kind of a clinical validation.
Okay. We also have a question regarding technique. How current do the control patients have to be? Are historical controls as impactful as concurrent?
From an FDA perspective, I would say yes. If you've got, for example, you have historical information that the rate of recurrence of breast cancer is I'm making up a number, 25%, and your new trial shows the rate of recurrence is 10%. that's comparison to historical population. The thing you have to be careful about is you can't be too historical, because if you're looking at the figures from 10 or 20 years ago, certainly treatments were different back then than they are now. So your historical has to be kind of a short-term historical to be acceptable.
And I think I would echo much of what Judy said from a payer perspective as well. If the historical control is simply to define a particular stage of disease, let's say by pathology, then it may not actually matter, how far back you look. If it's a stage 4 colon cancer, and you can recover the nucleic acid out of a 30-year-old sample, if all you want to do is show some sort of concordance between the molecular test and, let's say, the staging going backwards, the length of history may not matter so much. But much as Judy said, I think the FDA might care about, payers, I think, would also care about if you are trying to then use the clinical course or ultimate outcome of the patient that is the historical control as a demonstration of whether or not the marker was useful, the molecular marker was useful, then payers would probably care if the sample was so old that clinical standards of care had changed, because you might not be able to actually directly correlate the documented outcome for that patient that occurred 10 years ago with the outcome that they might have today if today's standards of care were applied.
We also have a question regarding some of the strategies you discussed. Where do lab-developed tests fit in the strategies you presented? Most of the new molecular diagnostic tests coming on the market are LDTs.
Well, from an FDA perspective, right now there is no absolute requirement, but I think and I'll speak for David, I'll put his hat on for a minute and say that if you're going to have to do a third-party payer type clinical evidence study, that you should think very strongly about adding in the FDA pieces to it, because in my opinion it's coming. Now, how the FDA will handle a look back on labs that have already established them, I can't tell. I think they might, but that's my opinion. But I think the smart thing to do is, if you're going to do third-party payer for your LDT, you should consider the FDA requirements too.
And I'll take my hat back for a minute and concur wholeheartedly with what Judy is saying. The fact of the matter is, these days, particularly as regards LDTs, payers they demand more evidence than the FDA does. I think that's pretty obvious by now. But as Judy said, given the way that the FDA regulatory winds are blowing, I think you would be foolish to only try and meet the needs of a payer right now because that's all you needed to do, when, before the test was fully mature in its market uptake, you might find yourself having to go back and do studies for the purpose of satisfying a post hoc regulatory requirement that had been applied.
Okay, I see we've come up on the hour, but we can take one more question. We've got quite a few more. I don't think we're going to get to them all. But for the last question, how do you see a molecular test which can replace, for example, a biopsy in terms of risk, evidence requirement, et cetera?
Well, I'll let David speak too, but from a third-party payer, I think they'd be very happy with it because it replaces a very expensive test. From the FDA perspective, since biopsy is number one, firstly that it is the standard of care and has been the standard of care for a gazillion years, and second, that it actually has human intervention. You take that specimen and you look at it versus a biomarker that you run through a machine and it says positive or negative. There will be a higher hurdle for that from the FDA perspective. You'll need to demonstrate in a large population of these types of patients that your marker produces the same results as the biopsy.
Now, from a payer perspective, Judy again has hit the mark in the sense that if indeed the biopsy and the pathology services required to make the diagnosis turn out to be more expensive than the test, just on the face of it, a payer would probably prefer the molecular test that actually replaced the biopsy. But I think the nuance that has to be brought out, which actually gets to something else Judy said, which is that if the biopsy has been standard of diagnosis for a gazillion years, the payers very realistically are going to be considering that there's going to be a period in which both are probably going to be done, okay? And in that context, then, the payer is faced not with a cost-neutral or cost-reductive diagnostic test. They're faced with one which is cost-additive for some period until the physicians become comfortable with the results of that molecular test that might have replaced the biopsy. So what that means, actually, is that the greater the quantity and quality of utility evidence that the innovator can provide for that molecular test, which would presumptively decrease that comfort period during which both tests would actually be performed, more receptive the payer would be because they would see a shorter period of practical dual usage of both kinds of tests.
Okay, excellent. Well, I see we're a bit past the hour now. We still do have more questions. I will send them along to David and Judy so that you can get those answers directly. I'd like to thank you both, David and Judy, for the wonderful presentations, and thank Precision for Medicine for their willingness to sponsor today's event. Thank you so much for taking an hour out of your day and spending it with us. I hope we'll see you at future CHI Global Web Symposia. And with that, I'd like to say thank you to everyone and have a great day.
Thank you, Elizabeth.
Thank you.